Awesome. Good evening, everyone, and welcome to Genomics Lite. This is our T-cells in focus session with our guest speaker, Megan. This is actually our last Genomics Lite session for this academic year, and I'm really excited about our topic today as well. So for those who've not joined us before, my name's Em. I'm the Science Engagement Manager at Wellcome Connecting Science on the Wellcome Genome Campus, and I'll be sort of hosting and making sure the session runs nice and smoothly. So I know many of you can see these slides before, but I'll do the housekeeping bit. So all the events that we host here are here to help everyone explore what genomics means for our lives and also for our learning, education and careers as well. So we're open to questions tonight. You can pop questions in the chat box or in the Q&A box for Megan, but we ask that you do so respectfully, and I'll be moderating the platform this evening to make sure it's a safe and inclusive space for everybody on the call. In case you've not used Zoom webinar before, you won't be able to use your cameras or your microphones this evening. So if you need to get in touch with us, you can use the chat box to say hello or to let me know if you're having any tech issues. Megan will also ask you some sort of open questions that you can put your answers to in the chat box. And if you've got questions for her about her work, her career, or anything on the slides, pop those in the Q&A box and there'll be time for those as we move throughout the session. We've also got closed captions available this evening. So if you want live captioning during the session, you can click the little CC box at the bottom of the screen. You can also move them around the screen to make sure they're never in the way. You can also get rid of them if you don't want them, but they're there if you do. So this is where Megan and I work, and Megan is coming from us live from the Wellcome Sanger Institute, which is always exciting. We are at the Wellcome Genome Campus just south of Cambridge in England, and yeah, that's where we both work, and everything that we do on site has something to do with genomics and DNA, and Megan will tell us more about her specific work in a bit. As always, there are extra learning resources available to go along with this session. They're available on our website, and I'll pop a link for that in the chat box in a second. But these are just to support sort of further reading around the topic of T cells and what sort of studying immunology looks like in the lab. For any teachers, educators, communicators that are joining us this evening, we've also got a Genomics Educators free Future Learn course launched at the moment. It launched at the start of May, but we're running it for the rest of June as well. So if you're interested, do join us on that course to learn more about teaching genomics. Lastly then, what our evening's going to look like. So I will very shortly hand over to Megan to hear more about her work in T cells. We'll then take a sort of break to have a Q&A around T cells and her research. And then Megan will share a little bit about her career journey and sort of how she's got to where she is today. And we'll have a bit more time for a Q&A then, but we'll be wrapped up by 5:45. If you have to leave early, we are recording the session and it will be available on YouTube and our website in the next couple of weeks. And that is everything from me. So I will stop sharing, and Megan, I will hand over to you.
Okay, great. So you can hear me okay?
Yeah, we can hear you great.
Great. And you can see that okay?
Yes.
Fantastic. Okay. Hello, everyone. I'm Megan. I'm a PhD student at the Wellcome Sanger Institute, as Em introduced. So I work in Gosha Trinker's lab, and our lab is focusing on genetic variation in a type of immune cell called T cells. My project involves editing the DNA of a gene associated with disease in order to find damaging mutations. So I'm a PhD student kind of doing genetics and immunology, which is both quite difficult, but it's also really cool, which I'll go on to explain. So let's start with a poll, just to get you guys interested. Can you name any cells of the immune system? I've just given one away, but please spew out some other ones.
So if you can think of any, literally any cell at all related to the immune system, pop them in the chat box and I will read them out for us. Awesome. We've got B cells, we've got lymphocytes, plasma cells, B cells, macrophages, dendritic cells, phagocytes, leukocytes, neutrophils, killer T cells, plasma cells. Cool. We've got a lot of memory cells. We've got granulocytes. I've not heard of those. So yeah, we've got lots and lots of cells.
Yeah. So that's loads. And I think I'm quite impressed, to be fair. I was kind of just expecting lymphocytes, but you are all correct, and these are all immune cells. So this diagram shows the different types of cells that you can have. So you've got the innate response, which are more broad and kind of your first line of defense. But I'm more interested in the adaptive immune system, where T cells and B cells reside. So just a bit more information about these. I'm not sure whether you know this before, but B cells and T cells are the two parts of the innate immune system. So what that means is that they attack specifically. So they're pathogen specific and will provide kind of a stronger response as well, and also a more memorable response. So your B cells mature in your bone marrow, whereas your T cells will mature in your thymus. The main functions of B cells is production of antibodies, which I'm sure you've heard all about. And well, T cells, what do you think T cells do? We've got a poll here for you to fill out. Do they just proliferate? Do they migrate to the lymph nodes and communicate with other cells? Do they function to kill infected cells? Or do they secrete these chemicals called cytokines, which communicate with other cells, or all of the above? Oh, apparently I can't vote. That's a shame.
I think that'd be a little bit rigged if you could vote. I actually think I probably can let you vote, but I think that might just give away the answer. Yeah, very quick response. We've got at least 80% going for all of the above.
Okay, great. So you realise how important T cells are, and they do have lots of different functions. So you can group them into killer T cells. So these are CD8 positive, and you can group them into helper T cells. So these are the ones that communicate more with other cells. And because they have lots of different functions, there's also a lot of things that can go wrong. So just to put these in the diagram, you've got your CD8 cytotoxic and your CD4 vector and your B cells here. So let's take a step back. What is a T cell? It's a type of white blood cell. There's about 25 million to 1 billion in the human body, and that depends on, it's a personal thing, it's also a genetic thing, it's also whether or not you're fighting an infection at the current time, or whether you're feeling quite run down. It's quite interesting how your T cell levels can vary just depending on your well-being. And they are key regulators of the immune system. And because of this, because they're so fundamental to the immune system, they're carefully controlled. So this is quite a busy slide, I guess, but I'll just talk it through. So they require 3 signals in order to be activated and to exert their functions. And so the first signal is provided by the joining of an MHC class 1 or class 2 protein. So this holds a peptide of the specific pathogen that you're fighting against. And that interacts with the TCR, so the T cell receptor. So that's signal 1 that's required. And if that's ticked off, you also need co-stimulation, which requires CD28. But then in addition to that, your T cells need to know exactly what kind of T cell they're going to form. And so they require cytokines here. Oh, sorry, I forgot I had these. Cytokines here in order to polarize towards a particular type of T cell. And so these exert slightly different functions and will fight certain different pathogens, even like more specialized. But that you kind of don't need to know just yet. So what could go wrong? What could possibly go wrong? Here's a diagram of the cell signaling. So once the TCR is initiated, I'm not sure whether you can see my pointer. Yeah, okay, great. So once the TCR is initiated, you have all these proteins that are activated downstream that activate more proteins, which then go on to increase the or influence the expression of the cell in order to exert their functions. And so I'm interested in a protein that is involved in this part, in the polarization or the getting cells to become a certain type of them. So this is the IL-2 receptor. So IL-2 is a key growth factor for T cells, and they require it to grow. It is a heterochimeric receptor, which is a fancy way of saying it's got 3 parts. So that's alpha, beta, and gamma. And what it does is that the cytokine IL-2 will bind to this, and then it will exert its signaling downstream, like what I showed you before. So we shorten this to IL-2RG because this is just long-winded. So why I'm particularly interested in this gene is because mutations in this gene are have been shown to cause immunodeficiency. So immunodeficiency is where your immune system is lacking so much that in this case, they have no functional T cells and no functional NK cells either. The B cells are okay, but you are severely immunocompromised, which means that you're at increased risk of infection. And if you're not protected, then you'll probably end up dying. And so this boy, you may have heard of the boy in the bubble. He was born in the 1970s with this, with a mutation in this gene. And this is an example of a very rare condition. It's rare because it's so, people will die. They can't exist like this. In fact, they can't exist unless they're in a bubble. So this was a NASA engineered bubble and a NASA engineered Earth suit, which allowed him to survive up to the age of 12, where he actually got an immune cell transplant from his sister in order to try and recover some of his immune cells. But unfortunately, she had a virus that was lying dormant in the transplant, which unfortunately then could proliferate regardless because he had no immune system. So it's very sad, but it's also a really cool gene. And what this kind of illustrates is that T cells, because they are so fundamental, if you have a loss of them, you will cause immunodeficiency. Whereas if you have too much of them, you might end up in something called autoimmunity, which is where you get an overactive immune system. So can anybody name any autoimmune diseases? There's quite a few. I think there's something like, I don't know, it's in the tens. So
yeah, I think it's one of those ones where a lot of like relatively common things that you wouldn't know, autoimmune diseases that happen, that just sort of are. Some great examples. We've got a lot of I don't want to say we have a lot of lupus, but there's a lot of the answer lupus, rheumatoid arthritis, MS, Crohn's disease, celiac. Another rheumatoid arthritis. An
interesting one actually, because it is an autoimmune disease, but it's very specific. So it's specifically against so gluten. So this is why celiacs can't eat gluten. It's because when they consume gluten, it will cause a modification on their cells in their intestine, and that's what causes the hyperactive immune system. But yeah, type 1 diabetes, Behçet's, I have no idea.
Yeah, I've not heard of Behçet's either. I think I'll be googling that one in a second. Psoriasis, MS again. Yeah, I'll do some googling.
Psoriasis, so that's focused on your skin. Again, another one I don't know. Pernicious anemia. So inflamed bowel disease, that's inflammatory bowel disease, but yeah. Allergies, it is a form of overactive, but less T cell mediated. It's more involved in the other counterparts. MS, Addison's, I'm not sure. I think
I have
that's
not
when
heard
you've
of that
got
one.
too much. Is that when you've got too much steroids? I'm not sure. Anyway, you're doing great.
Yeah, we're all learning stuff
And
today.
you kind of made me think that I need to read up on some things. So yeah, you've got multiple sclerosis, which is based in like your neuronal system, rheumatoid arthritis, which is inflammation of the joints. You've got inflammatory bowel disease, which I'm particularly interested in, which I'll come on to later, which is obviously based around your gut. And then psoriasis, which is also your skin. So you can see why I'm interested in T cells. It's very cool. But how am I investigating these? And here at the Sanger, we're very focused on genetics and particularly modifying the genome. So we do this through a technology called Crispr, which, if you want the long-winded, which personally I don't think it means a lot, clustered regularly into space for short palindromic repeats. So that just refers to the fact that it will recognize a certain part of your DNA. And what it does is that it allows us to target and specifically edit DNA. And in my case, I'm using this to test all possible mutations within the gene. So the gene that I described, IL-2RG. So just as a bit of a background, CRISPR is composed of a Cas9 enzyme. So this enzyme, the blue here, forms with a guide RNA. So this is a specifically structured piece of RNA, which will then directly target whatever it's designed to target here, and it will locate to the genome and where it will specifically bind here and induce a cut. And so there's various ways of introducing edits that you want through this thing called homology directed repair, where you provide a template called, there's also prime editing, there's also base editing, there's so many, and there's one that comes out every single week, it seems. So what is a mutation? I just want to check that everybody knows this before I go on anymore. So could anybody provide even just keywords of what it is? A random change, yeah. A change in base sequence, yeah.
I think just because everyone's got so many, hit so many great ones earlier, if anyone knows sort of types of mutation as well, stick those in the chat box since we've got so many specific disease ideas earlier. See if we know any others. So yeah, we've got general sort of the ideas of change in base DNA. So we can have an addition and deletion, yep. All frame shift mutation, we know some, very specific.
Yep.
Substitution, yep, awesome. I think we've all got a good base understanding what mutation is, but I know that you're going to go through what those are in a second as well.
Great to know that everybody knows. But yeah, you're correct. It's a change in our DNA sequence. And this does happen naturally. But we have repair mechanisms in our cells, which mean that it normally doesn't progress. And so cancer is an example where a mutation is caused, but the mutation can cause the cells to override that normal safeguarding mechanism of killing the cells. And environmental factors, such as UV, smoking, also bacon, I hate to say it, increases your risk of colorectal cancer about 4-fold. I'm personally vegetarian. So this can increase your risk of them. So that's why we wear sun cream and become vegetarian and don't smoke. So just as a recap as well, so we have our DNA, and this is transcribed to RNA through various enzymes. And then it's translated from RNA into amino acids, which can then form a protein. And so you can see that, sorry, we'll skip on to the next one. So you have the original sequence, and a point mutation can cause one specific nucleotide to change. Or you can have a substitution. You guys were all great at describing all these earlier. Insertion is particularly damaging in the fact that it will shift. And so when you have a codon, which will code for a specific amino acid, if you shift it by one, all the downstream amino acids are going to change. And deletion quite equally has the same effect, and also inversion. So these are kind of the smaller things. But as I said, this could have the effect that there's no protein expressed. So with a change in DNA, you have a change in RNA, and with a change in RNA, you have a change in protein. So this can cause the protein that is encoded by the gene to not materialize at all. So no protein. You can get a change in protein activity. So the protein can become dysfunctional and no function, or it can become hyperactive too. That's also particularly damaging. Or you could also generate a completely different protein. There's actually a really cool example in the lab where we use green fluorescent protein. So this is a gene from a jellyfish, and it's used as a reporter for a lot of things. But interestingly, if you change one nucleotide, it will turn to blue fluorescent protein. And so it's used as a way of testing kind of your efficiency of editing. So in addition to point mutations, you also have complete chromosomal alterations. And this is particularly seen in cancer and also cell lines. So cell lines are immortalized cells, which are normally derived from cancer patients. And probably one of these examples is the HeLa cell line. If anybody fancies reading a really good book, it's The Immortal Life of Henrietta Lacks is really interesting. Basically, it was this woman in the 1940s who, I think it was cervical cancer, and her cells were taken, and they continue to grow even now because they just can't stop dividing. They've got so many mutations. So anyway, sorry, that was a bit of a sideline, but what we can use spectral karyotyping or chromosome painting in order to investigate chromosomal alterations. So here you can see a nice healthy genome where there's 2 copies of each of the chromosomes. And so like yellow is chromosome 1, red is chromosome 2, yeah, further down you can see other ones. So that's normally. However, this is the cell line that I'm currently working with. So can anybody point out particular things that are wrong with it? I think first of all, the copy number. Oh, I've just seen Em's given a short read on HeLa cells, very good. So there are, yeah, so not identical. So they're not identical because they have translocations. So this you can see in chromosome 5 and also chromosome 13 that there's translocations where the chromosome 13 is swapped with chromosome X, which causes this abnormal chromosome. And there's a Philadelphia chromosome, which I can't remember which translocations it is, but it causes cancer. So yeah, this is a result of always being able to proliferate without having those repair mechanisms in place. And so back to what I'm doing, what we know is that certain variants within the gene that I'm interested in are associated with disease. As in, if you inherit a variant here, say the green represents an A to G, this could be seen in somebody in the clinic. And so we know for diagnosis purposes that this variant is going to cause disease. But we rely on the fact that somebody comes in with that disease. And so that delays diagnosis, and it also reduces, if we know more, we can figure out particular portions of the gene that we could target with therapies as well. And so what I'm trying to do is investigate all other possible mutations and find these arrows here. And so what I'm doing here is saturating the gene in order to see their effect and finding out whether these mutations are associated with immunodeficiency or whether they're associated with autoimmunity too. And so this has been done for the BRCA1 gene. This was the first gene that this technique was used for. So this is the breast cancer gene, or the Angelina Jolie gene, because she famously inherited it and ended up having a mastectomy due to her increased risk, because the majority of women that have this mutation will develop breast cancer by the age of 80. And so what they did in this technique was they did introduce every single variant into the gene. And as you can see in this diagram here, this is a structural representation of the gene, the protein itself. So you can see that the red has the most deleterious variants enriched in it, whereas the white is, if you have a mutation, it probably won't cause anything. It's not pathogenic. And so that's a brief summary into my project. I think it's really cool. But what does this mean in my day-to-day? So I thought I'd just give an overview of what a day in the life of a scientist actually consists of. So I tend to start quite early, just because I'm a morning person. I know people that start later. It's not necessarily required. So I check on my cells. Em found this quite funny when I was running through it with her, that I check on my cells and feed my cells like somebody feeds their dog. But because they're continually growing, they're not going to stop. They're only going to stop if you stop feeding them. And so what we have is culture, so media, which is a liquid which contains all the nutrients that it possibly requires. And we have to make sure that they have enough media in order to survive. If not, they're not going to be happy. And then after I've checked on my cells and fed them and given them any cytokines that they require, I typically read a paper, because reading papers to make sure that you're on top of the literature is incredibly important in science. There's a paper out all the time. It's really hard to keep up, but there's also ways that the lab we support each other by having journal clubs where we take it in turns and present other meetings, present to each other a particular paper that we're interested in. And then this is a typical Monday, we'll have a lab meeting. So there's 15 lab members in the Trinker lab, which is quite big. I've also been in labs where there's only been 4. And there's pros and cons to being in big labs versus small labs, and I'd be welcome to discuss if anybody was ever at that stage yet. And here we typically take it in turns, discussing our project, trying to get input from other people so that we can get advice. The general thing with science is that, particularly at the Sanger, we're very collaborative, which means that if you have an issue, you want to share it with other people to get them thinking about it. And that's a lot more successful way of progressing than keeping things to yourself, which is quite prevalent in academia, because people are worried about somebody stealing their idea. And then I would go and do some admin like every other job, answering some emails, checking on orders. Then team lunch at like 12. And then I'll go on to do something called flow cytometry staining. So does anybody know what flow cytometry is? I'd be quite impressed if you do, because this is something that you only really do at university. Well, not even university, because they're expensive machines.
Yeah, I was going to say the machines aren't cheap. They're not ones that you really get anywhere near until very high up.
And when we say expensive, we mean like £250,000.
Yeah, nothing in science is cheap. So if we say expensive, it's
yeah,
silly money.
Exactly. In which case I'll explain. So flow cytometry is a really cool method that allows you to see what proteins are expressed on your cell or in your cell. And what you do there is it composes of, so you stain them first of all. So what you do is if you have a protein of interest here, so say this is CD4 on my T cell, I will stain with an antibody which is conjugated to a fluorophore, so something that fluoresces when you expose certain light to it, and then it will emit a different light. And then we run this through the fax machine. So this is a picture of the machine that costs £250,000. And the cells go up in here, they get shot, like lasers get shot at them, and then you analyze the amount of scatter that comes off them in order to see whether or not they have this protein or not, or if they do, to what extent. And this is on a single cell basis, so it's really quite crazy. And you can also do this to sort. So if you're interested in cells that are CD4 positive, then you can say, okay, put all the CD4 positive cells into this tube and put all the negatives in another tube. And then you get something like this out. So I here stained for my protein of interest, IL-2RG, on my cells. And this is my unstained control. And you can see that when I stained and added the fluorophore, you can see that my cells do indeed express my gene, which is really cool. And so I've been seeing whether my mutations influence the expression of that gene. So then after flow cytometry, I typically do the analysis, and from start to finish is probably about 2 to 3 hours. I think I've been a bit optimistic here because I wanted to show off, but realistically, everything takes 5 times longer.
We can call this a super productive day. This is like you've had lots of coffee, you're on it.
This is caffeine, sugar, everything. So then I'll write up my results and make sure that they're all neat and tidy, because I'm all for memory as well, which means that if I don't write it down, I'll forget about it. And it's very important that you do write down exactly what you do, because everything's so exact. And then I'll plan my next experiments and my future plans and prepare for the next day. Typically asking people for advice around here as well. So I think at that point, it's probably a good point to ask for any questions. I don't know how we're doing for time, but
absolutely spot on time. And actually someone put a little explanation of what they understand from flow cytometry, and I think they were pretty much spot on. It always amazes me how much lasers are involved in biology. So they put a laser illuminates a single cell stream and characterizes them by the transmission observed, like cell size and other stuff. I like the other stuff that covers most bases.
That's a really good answer. So admittedly, I didn't show all the kind of plots, but you have this thing called forward scatter and side scatter, and that allows you to see the cell size of the cells. And so you get this dot plot instead. But I thought I'd just keep to the peaks. But that's really good.
So if anyone's got any questions for Megan about anything we sort of talked about before we move on to the sort of careersy type portion, then pop those in the chat box or the Q&A box, and I'll pull some of them out. I wonder, I know that you're sort of in your first-ish year of PhD, but have you found anything exciting so far that is promising, or is it just a lot of testing stuff out at the moment?
It's a lot of testing stuff out, and PhDs are typically, like, it's quite known that you don't get a lot of results from your first year. You're typically just training, amongst PhD students, we have the general consensus that you have to make every single mistake so that you won't make it again, which is kind of like a brutal truth, but you know that you're not going to make it again if you do it. So there's been times where I've accidentally thrown away my cells and accidentally used the wrong buffer. What else have I done? One time I used 10x PBS. So PBS is a phosphate buffered saline basically it's equivalent of, like, it's water plus salt, so that the cells, when they're in it, don't explode. And usually you use 1x, and so I use 10x, which is 10 times more concentrated, and it causes the cells to shrivel and die, which was not great. But it's all part of the learning experience. So question, how did you choose the topic for your PhD? Good question. And actually I'm coming on to that. So I'll put that to the one side for the moment. Is there any other questions?
Someone asked for a quick recap on flow cytometry because their internet dropped. So if you could, give us another two sentence what flow cytometry is.
So I think, is it Dennis?
Yes.
Provided a good explanation. So what you have is that, if I go back to this slide, this one. So what you do is that you have a protein that you're particularly interested on the surface of the cells. So in my case is IL-2RG, and that is tagged by an antibody which is attached to fluorophores. So what this means is that when the antibody binds specifically to the protein that you're interested in, it will fluoresce. And that is what you detect on a flow cytometry machine. So the cells run through this machine in a series of tubes, and in the tubes then they get lasers shot at them, and in the way that lasers then bounce off, that is detected, and you can say explicitly whether the cell, that specific cell, expresses that protein. And yeah, like I said, you can also use that to sort as well.
Someone sort of followed up saying, so is it test to see if the protein is present?
Yeah.
And it gives you sort of an amount as well, I guess.
Yeah. So it's to see whether it's present, but also the intensity. So if I found, if I'd given them some kind of stimulant or some inhibitor, if you had an inhibitor, you'd have less protein. So it would be further to the left, so less intensity. And if you gave an inducer, then you'd have a higher intensity of the protein.
So I'm guessing that you're hoping to see, once you've done your CRISPRing, that different mutations are going to shift that, right? And it's either going to be less protein or more protein. But I guess it doesn't tell you, I guess it just sort of tells you that there is, like, there's more or less, doesn't sort of tell you how or why, but it gives you that, more or less protein.
Exactly.
So someone's asked, in autoimmune diseases, what determines which part of the body is attacked? Is it the surface protein on a particular part of the body?
That's a really interesting question. And I think probably the best way to explain it, of course, there's a lot of things to do with autoimmunity that isn't fully understood. But the general idea is that you have antigens on all your cells. So not just pathogens, but all your cells will contain an antigen which will say, I'm this cell type, and I express this protein. And that also links to their function. So for example, T cells will express IL-2RG because they require the IL-2 signaling pathway. But quite equally, these antigens on your own cells can induce an immune response. And so that's where you get autoimmunity. I think MS is where you have attack on the myelin sheath. So the myelin sheath covers your neurons, and it has a specific protein signature, so a particular antigen that is then recognized, and your immune system attacks it as if it's a pathogen, which causes inflammation and damage. And yeah, it's really interesting. But also, so there's been a lot of work to do with viruses. So a bit of COVID, but also if you've heard of Epstein-Barr virus, they found that actually Epstein-Barr virus so a lot of MS patients have been infected with Epstein-Barr virus, not so much as it will cause MS, because also a lot of people healthy have been infected too, but they think that there's a certain genetic predisposition that if you're then infected with Epstein-Barr virus, then it can modify your immune response and you can develop MS against it. So the way that the immune system is in careful balance, and then if it gets hyperinflamed towards a certain antigen of your own immune system, of your own tissues, then that's where you can get autoimmunity. Do you think in the future that you'll be able to cure autoimmune disease? I hope so. There's a lot of work that is being done, and I think it also depends upon your sorry, I think somebody's just running for the bus and making the whole floor shake. I think it depends on the type of autoimmune disease. So, for example, celiac disease is arguably, I don't know, I'm not an expert, but one of the easiest ones to treat, in that people just have to avoid gluten. And so therefore you won't get that modification on your cells, and then you won't get them to recognize it. But there's also other ways of reducing the inflammation in the body. So a lot of the therapies are focused around like steroids and reducing your immune attack. But then you are also at an increased risk of developing infections. So, and I think the main issue is that there's so many different causes of autoimmunity that we haven't got a specific therapy for it. But
yeah, it's kind of like cancer in that it's like it's a category of stuff rather than a single disease by itself. So I guess, you know, the more we can learn about the immune system in general, the better chance we've got of being able to do more things against autoimmune diseases in general. But because it's such a broad category of disease, it's not like there's unlikely to be like a one size fits all. It'd be great if there was, but probably unlikely.
that's a good example.
Yeah, I think if we move on to your sort of career journey aspect, and then we've got time for more questions after that as well.
So I'll go back to the question, how did I pick my topic for my PhD? So I think, did I think that I would end up studying what I am now? No, in full honesty, I actually hated genetics at university. So I studied biomedical sciences at the University of Bath. This is me, by the way, I like cycling. And as I said, I hated genetics. I found it really difficult to think about. It's quite abstract and it's quite conceptual. It's a bit like physics and maths. It's more that side of things than like clinically relevant. And in fact, I was more interested in pathology. And this was helped by the fact that I had a really good lecturer, Professor David Tosh, who had this really cool way of lecturing as well, which was very stripped back. So he wouldn't let you get your laptop out. And in fact, he just wanted your full attention during the lecture. You could only take notes by hand, but otherwise he would write everything on the whiteboard. He never had a PowerPoint. And I think his style is really good because sometimes you can get very caught up in memorizing, particularly in biology, because there's so much to memorize. But actually, if you just listen and enjoy kind of the science, then I think you'll learn a lot more than just taking notes and being stressed. And so I think it sounds cringy. Oh, I've got cheesy lessons here, so when I was putting this together, I was like, what would I want to have heard? So, and I would have wanted to hear how people's career progression has also like what lessons they've learned, and so that I could learn from those lessons. And so I think cheesy lesson number one is follow your passion. And I did indeed follow that passion of pathology. In those lectures, I was particularly interested in inflammatory bowel disease, and particularly interested in like the immune system of the gastrointestinal tract. So I've got a slide here of as a brief facts of the gastrointestinal tract. So I was particularly interested in the microbiome, which consists of 100 trillion bacteria and it weighs about 2 kilos in your gut. And there's about 10 times more bacteria than human cells. And in fact, 70% of our immune system is actually residing in our gut. So that means all our T cells are resident in this tissue. So I found it really interesting because I went to work experience at a pharmaceutical company called UCB. So I asked around and I knew that I wanted to go into science, but I just wanted to see what it was like working in the lab. And it was actually a discussion at lunchtime where we were discussing the difference between identical twins. So identical twins have exactly the same DNA, which makes them really cool things to study. If you know an identical twin or you are one, that's cool. But what makes them different? Because some of them will manifest disease, some of them won't, some of them develop different, so much as different freckles, that kind of thing. And it's all environmental. And part of the environment is your microbiome, because this is shaped by everything that you do, everything that you eat, everything that you drink, everything, the amount of exercise that you do, everything. And so I found it really fascinating because it could be potentially a way that you could reduce your risk of getting certain diseases by modifying your diet and modifying your lifestyle. I liked the flexibility in it. So I followed my passion, and Bath was also very keen on doing placement years. So I spent my third year at GSK, which is a pharmaceutical company. And here I did a project on inflammatory bowel disease, which was really cool. And it allowed me to see the industry side of things. Because industry are obviously focused on developing therapeutics, it was very clinically focused rather than just doing things for the fun of it. I say for the fun of it, more fundamental. And so this comes to cheesy lesson number 2, was that you should always try new things. And I could have just plowed through my undergraduate project, but I gained a lot more from putting myself out there and doing a different thing in a different place and experiencing something different. I think it highlighted that I didn't want to be in industry, at least not for the time being, because I found it quite slow paced, which was frustrating, and I wanted the freedom to do what I wanted. But what it did do is that it also exposed me to different people. And interestingly, somebody that I was working with at GSK was actually on my interview panel at the University of Cambridge. So that was really cool because he knew that I was interested in studying IBD. And this project here, I was looking at the genetic variation associated in T cells associated with IBD. So you can see how I gradually got into genetics. And so then it was a bit complicated, but James, my supervisor here, he wanted to move. And this is quite uncommon really, but I didn't want to move. And so I was looking for a different project. And in this project, I was exposed to Gosha Trinker's work at the Wellcome Sanger Institute. And so I reached out to Gosha and said, do you have space for a PhD student? And so that's how I've ended up doing genetics, from a windy road of following my passion and trying new things. I'm now in the project that I am, and I'm very much enjoying it. So I do think there's something good about planning and having an idea of exactly what you want to do. But I think also if you follow those key rules, then you never know where you're going to end up, basically. That's my life lessons. And so key take-home messages I'd like to go on to here is that T cells are super cool. They're involved in immunodeficiency and autoimmunity, and my project is involved in identifying mutations that push them to disease. And in terms of key take-home messages of life, follow your passion and try new things, because you don't know where you're going to end up. So sorry about the cheesy ending.
We love some cheese. Awesome, thanks so much. And yeah, you definitely covered the why did you choose your PhD topic? So if anyone's got further questions for Megan, pop them in the Q&A or chat box. But yeah, I'd reiterate the I think especially in science that it's great just help for the thing that you'd like to do next, but actually science, but sort of biology and genomics is so skills based that actually you might do one project that you like because of the topic, but pick up skills that you then do the same skills on an entirely different project. So you kind of don't have to worry too much about just sort of pick up skills and sort of focus on that and what you like, and it'll all work out. Quite a practical one. So what are your thoughts on this? So I want to maximize my chances of getting into university. What are ideal things to put on personal statements, especially for biomedicine? So also, we're not careers, we're not university advisors, but if you've got any top tips that you remember or would like to share.
So I think the main thing is that, so obviously if you want to go to university and study this course, you must be interested in it. And so you want to show that in an interview, but you also want to have experience which shows this. And so by me going to UCB here was really good for just showing that I was really interested in it and really wanting to learn more. So I think things that obviously grades are important, but I didn't get the best grades, particularly in my undergraduate. I notoriously got 0.02% into a first class degree, but safely. Anyway, so I think, do things that show your enthusiasm. So try and get work experience if you can. I know it's difficult and it's very much who you know, but a bit of persistence is kind of key there. But even then, making things for yourself. So I don't know, doing things on YouTube, or starting little science clubs, start a journal club or something at school that makes you stand out from the crowd, because I know it's so competitive. But you just want to show that you're enthusiastic, and this is the thing that you want to study for 4 years or 3 or 4 years.
I'd sort of second that and say that you want to show that you've not just gone to school and gone home again. You've and I know that I know that that's enough, and I know that should be enough, but you kind of want to show that you've done stuff above and beyond. So I know that work experience is particularly hard to come by at the moment. There are increasingly places that are offering sort of online work experience packages. Obviously they're not the same as the real thing, but they're still going to show that you're putting yourself out there, you're trying. So if you're UK based, I'd recommend Springpod. I've not done any of their programs themselves, but they've got really big companies like BA and Data Lite, Lite do sort of virtual work experience packages through them. So that'll just sort of show that you've really sort of tried. There's places like Nuffield that do in-person research placement. So hopefully if you're at high school, sort of teachers and stuff can point you in those directions as well. But virtual work experience is coming a little bit easier to find. So hopefully if you're in a position that you can't do it in person, that might be a starting point.
Mm-hmm. I think as well, something I just thought about was books, reading. Work experience is hard to get, but ordering a book is really useful. And one author that I really like is, what's his name? He works at the University of Manchester, and he has very public outreach community. I'll have a think. It will come back to me. That he did the compatibility gene, and Yeah, oh, I can't remember, what is it? I want to say it's
Daniel Davis.
Daniel Davis.
There we go. Some
That's
Googling got me there.
Yeah, there you go. Not Matt.
It had it's Daniel M. Davis. We'll say that his middle name is Matt. Someone's put in the chat box a similar sort of university-related question, so I'll stick on that for a second, saying, I'm having a hard time deciding whether to do nat science, biomed, or biology at uni. What made you choose biomedicine specifically over other biology-related degrees? Was there anything specific, or was it you liked the uni, you liked the course, so you kind of went for it, or was there something about it being a biomed degree that caught you?
So I'm going to provide advice here that I didn't follow. So I think the advice is, if you, so Nat Sci is a lot broader, or at least it was at Bath. So if you're kind of interested in not just biology, but also like physics, also chemistry, then do that. If you know for sure that you're more interested in humans, then do biomed, and that's kind of why I picked it. And biology, I think if you're open to humans and plants and like viruses and bacteria, then go for that. I think it depends on whether or not you know that you're interested in that level. I personally hated plants, and so I was like, I'll get rid of that. But actually, so that's my advice, but I actually applied for medicine at university. And so on the UCAS form, you could, or at least back in 2015, you could apply for 4 medical placements and then 1 non-medical placement. And so Bath was actually my only offer for biomedical sciences. And so I thought, do I take a year out, do some work experience and reapply for medicine, or should I go ahead and do this? And so I read more about biomedical sciences and about the course, and I was like, I think I could try this. And then I always thought that as well, if I wasn't happy, then I could do graduate entry medicine. But actually I was very happy with research, and so I decided to stick with that. So yeah.
I'll just add on the degree title thing, because it's something that I got bogged down as well with at uni, that I think it's really easy when I had like a stack of 20 like university prospectus, and they all call their courses something a bit different, and actually you kind of, unless you know you want to go into like medicine, which is a sort of different kettle of fish, or like dentistry, if you're in that sort of biology sphere, a lot of places will call their degrees slightly different, but actually you want to look at the courses inside the degrees. So I applied for like biochemistry, molecular biology, cell biology, which were all different names, but actually the courses that they did inside the degrees were all pretty much the same. So I'd say try not to get bogged down too much in the specific like degree title. And then I'd also look at how flexible the degree path is, and that's sort of at that uni. So some unis you start and that's it. Those are the courses you're doing. You've got no sort of choice in your courses. You have to do these ones, and you've not got much choice of sort of changing. I chose to go to Scotland, where I didn't have to choose my actual degree title until the end of my second year. So I could be very, very flexible. So I'd say sort of take that into account as well, that it's not all on your degree title, and that every uni is slightly different. So try and sort of see past the degree title a little bit, and sort of pick the actual courses that sound interesting, rather than just that sounds like a good sounding title. And if in doubt, the more flexible the better, would be my last sort of word on that. Mm-hmm. Back on your PhD a little bit more, someone's asked, what's your favorite part of your PhD? So you've said about what a day in the life looks like, but what's the best things about doing a PhD?
For me, I like the freedom of it. I have many friends like in London on these like corporate jobs, getting paid loads, and having a work-life balance. But actually, I have so much freedom in what I do. So if I find an experiment that I particularly want to do, then it's a case of asking my supervisor what they think, more for advice than anything, and then being able to do it. And my project is mine, and I can decide which avenue it goes down. So like I said, I'm more interested in like clinical things. So the project that was proposed to me initially could have been very fundamental genome editing, but I changed it to more clinically relevant by picking a gene that is involved in disease. So I think that's probably my favorite part, the fact that I can decide what I do. Also, you're a bit more flexible with working hours, just the fact that, well, at the Sanger it's a bit different because there's buses and you have to get them. But I have PhDs, other friends in town that work whenever they want. They do work at weekends, they do work in the evenings, but it's flexible, which I think is quite rare in a job.
On the flip side then, what are maybe some of the harder parts of doing a PhD? Because it's obviously not everyone becomes a doctor, it's not a walk in the park. So what are some of the slightly more challenging bits?
It's not easy. And I think things don't work. And so I think it's more the emotional roller coaster of doing a PhD of, yes, it worked, to, oh no, it didn't work. And then you can have very low for a long time, and then something can work and then you love it again. So I think it is kind of the emotional roller coaster, and like resilience is particularly important in order to pick yourself up and continue on, because it's 4 years. So it's a bit of a marathon. So what A levels did you do, and any revision tips for them? So A levels, what did I do? I did biology, chemistry and maths because I wanted to do medicine, and they were the things that needed, but also the ones that I was best at and the ones that I was most interested in. I also did French at AS level because I just enjoyed French, and I like French food. So I think I remember like studying very hard for particularly for biology, because it was such a memory game. And I think at secondary school it is, you're kind of taught that you have to memorize. And I do wonder, if I step back to student life, if I did just kind of absorb it more and enjoy it more, whether I would have done better or worse, I don't know. But I think I think when you're interested in, you actually want to study. So I think making yourself interested, if you can, and picking subjects that you are interested in is more important than topics that you're kind of being told to do. Your parents might agree otherwise, but I definitely think pick what you enjoy. And revision tips, I did a lot of, I'm a very visual and doodling kind of learner. So I used to make so many pretty notes, all color-coded and highlighted. But I also remember doing study sessions with my friend who was also doing the subjects that I was. And so we do study sessions where I think recall is really important. So we'd spend an hour studying, writing notes ourselves, and then ask each other questions, so that we kind of mimic doing past paper questions. And then obviously past paper questions are really good as well. But yeah.
I'll add to, I mean, my advice is a lot older than your advice, but I'll say, to start with, A levels are hard. I think that that's worth acknowledging. I've done A levels and 2 degrees, and I'd still say that A levels are really, really hard. So accept that, and if you're going to do them, fab, but they can be really hard. And then I'd say, at the end of the day, to an extent, you do have to sort of do the learning for the exam rather than just for enjoyment, which kind of sucks to an extent, but it is sort of how it is. And past papers is,
you can just disregard plants if you're not interested in them.
Not at A level. You can do that at degree, but not at A level. but yeah, past papers and sort of knowing what the exam wants you to write is not a way that we should have to learn, but definitely is the way to get through A levels. Knowing what, when it asks a question, what is it actually looking for? What are the keywords? What are the buzzwords? And just going through past papers as like and spotting patterns. So I remember in my biology A level when there were specific words that it would use, and it always wanted you to talk about X, Y, and Z. And so I kind of just like had that in my brain of like almost translating the question into what makes sense for you. And yeah, past papers is kind of the way of getting there on that. Awesome. We've got one last question at the moment. What was your favourite and least favourite aspects of doing biomed at uni?
So university is a whole different ball game to school. And I think I liked the fact that you're being taught by experts in the field, because they're very passionate about what they do. And that's really interesting. I always loved going to lectures, which some people didn't. And so I think probably the lectures were one of my favourite parts. I think the least favourite aspect, I didn't enjoy studying for exams, because nobody enjoys studying for exams. But it is kind of like part and parcel that you have to do it. But then, like I said, like finding people that you can study with, I think some of the best things about university is that everybody's in the same situation. And so we're all, like, you have study groups and you all work together studying the library. And so if anything, if you're not studying, you're kind of being left out. So yeah, I think like also just finding new friends and trying new things was really good. Like I definitely completely changed at university and loved it. It was great.
Awesome. And someone's asked, because you said that you would have done medicine if that's what you applied for. Someone's asked, would you now do graduate medicine, or are you very happy feeding your cells?
I always romanticize it a little bit and think, oh, it would be really cool to do medicine. But actually, I now know, I think I went into medicine a bit because I knew that I was really interested in like the human body and all the different things to do with that. But I think it was a part of, I wanted to do medicine because it was the only career that I knew that I could do that for, like study the human body in. And so I think now I realise that there's so many other careers that you can do that are open, that still allow you to do that interest with actually a lot more perks, because working in the NHS isn't particularly fun. It's very stressful and you kind of have to evaluate what you want in life. And I think it's really hard that people at your age are having to decide whether they want to do a career, a particular career, because I think you kind of have to try it some more. But that said, like I have friends that are doctors and they love it. So when I talk to them, I'm like, oh, maybe I should. But I think now I realise that there's so many other avenues to go down.
Yeah, I think a lot of people, and not that they would regret it, but a lot of people that are going into medicine know that they want to do something in biology, and there's just a sort of lack of information about different careers, which is why we do sort of sessions like this. And there's also all of those careers that like are in a hospital, but in like a hospital lab. So I've got lots of friends from my degree that work in a hospital, and the samples that doctors take and things that doctors do all get handed to them, and they do all the tests live in the hospital. And I think that's a really cool sort of in-between job as well. So yeah, I think there's just so many different biology jobs that you don't have to be a doctor to sort of work in medicine.
Exactly.
Awesome.
And I think I'm going to treat more people if I develop a drug.
Yeah, and it sounds for you, especially like you really enjoy the freedom of of working in a lab, which obviously if you get a patient, you can't be like, oh, I don't fancy treating that one today. So yeah, there's different perks to different jobs. I think that looks like all the questions we've had in today. So thank you, Megan, so much for joining us. I've definitely learned loads about flow cytometry in particular. This is actually the last of our Genomics Lite series for this academic year. We will be coming back in September, October for a sort of new batch of talks. So on the screen is a link to a SurveyMonkey. This will also come out in the Eventbrite follow-up email that you'll get maybe tomorrow, maybe early next week. But on that survey, we've got sort of questions at the end about what other topics you'd like to see. So if there's anything that you'd like to know about specifically within sort of genomics, DNA, and sort of biomed topics, let us know on the survey, and we'll see if we can source some speakers for next academic year. But thank you very much again, Megan, and thanks everyone for joining us. And yeah, hopefully see you again in the next one.
Thanks. Bye.